Hi, my name's Cameron Burke, and welcome to this Kaplan MCAT Science Review for control of gene expression in prokaryotes. This video covers basic operon structure, and then gets into the specifics of two different operon systems you might be asked about when you take the MCAT: inducible systems and repressible systems. The punchline for this video is to know what the important players in prokaryotic gene expression are, what they do, and how they work together. Right now, words like lac repressor and lac inducer might seem like they're made up, but by the end of this review, you should be able to answer questions like this one about operon function, so keep that in mind as you work through the video. The bedrock of prokaryote gene expression is the operon. Operons are basically just a team of several genes with closely related purposes that originate from separate pieces of DNA, but then stick together to be transcribed in close succession. The reason these genes are glued together into an operon is that they have the same job. They're in charge of regulating gene expression. This particular arrangement of genes is called the Jacob-Menard model. It traditionally has some structural genes, an operator site, a promoter site, and a regulator gene. The structural gene codes for the protein that needs to have its expression regulated. This is the business end of the gene because this is what actually produces the gene product. Upstream of the structural gene is the operator site, which is called a site and not a gene because it doesn't get transcribed. It's a piece of DNA that our repressor proteins could stick to if it's needed. Upstream of the operator site is the promoter site. It's just a place for RNA polymerase to stick onto the operon. Finally, the regulator gene is the last in the sequence. The regulator gene is the party pooper of this whole operation because its only job is to code for the repressor protein that stops the gene from producing its product. Operons always have the same regions, but there are two kinds of operons that work in slightly different ways to know about for the MCAT. The first type is the inducible system operon. Inducible systems are usually in the off position, so gene products aren't really being produced, and they have to be turned on to create more gene products if that ends up being necessary. The repressor protein coded from the regulator is typically stuck super tightly onto the operon at the operator, and it blocks RNA polymerase's way so nothing can get made. To circumvent this, when more gene product is needed, the inducer comes in to intercept the repressor protein and bind onto it before it can reach the operator. Then the repressor is too busy dealing with the inducer to get in RNA polymerase's way, and gene product is created. Inducible systems can be called positive control mechanisms because a new thing has to be present for something to happen. The result of the whole process is production. We'll cover what a negative system is when we get to repressible systems shortly. The inducer coming to get the repressor before it can block RNA polymerase is pretty similar to competitive inhibition, so remember that similarity for test day to help you with this whole process. A classic example you might see on the MCAT is the bacterial gene that produces lactase. Bacteria can digest lactose, but it's more energetically expensive than digesting glucose, so they only do it when they really have to. Most of the time the lactase gene is turned off, but occasionally it gets turned on, so it's perfect for an inducible gene. Repressible systems are sort of the opposite of an inducible system. While inducible systems need to be turned on, repressible systems need to be turned off. They're constantly making their gene product unless they need to stop for some reason. Repressible systems work by having the regulator gene code for a repressor protein that can't work by itself. The repressor tries to bind, but it can't, so RNA polymerase gets to do whatever it wants in terms of gene product making. However, if the bacterium needs less of a gene product, the repressor has a buddy protein to come and help it, called a co-repressor. The repressor and the co-repressor bind together to make a team of repression that can stick to the operator, get in RNA polymerase's way, and slow or stop gene production. Repressible systems are classic examples of negative control mechanisms because the usual state is producing something, and when the system is working, there's an absence of gene production. The lack of something being produced at the end is what makes it negative. The trip operon is one common example of a repressible system the MCAT might ask you about. When the environment is super high in tryptophan, it actually acts in a negative feedback mechanism. The tryptophan molecules are the co-repressor, so the bacteria stops making tryptophan by itself when there's a ton of it lying around to stick to the operator, because you shouldn't waste energy making tryptophan that you can find in your environment. Now that you've had a chance to review different kinds of operons, check out how the learning goals are going for you with this question about genes in the operon. The correct answer is C. The repressor binds to the operator site so it can stop RNA polymerase from making gene products. To recap, inducible systems are positive control mechanisms because something is made when they're working. When more gene products are needed, an inducer comes along to bind to the repressor protein, allowing RNA polymerase to get on with its job. On the other hand, repressible systems are examples of negative control mechanisms. Transcription's usually going on, but if it needs to stop for some reason, a co-repressor comes in to team up with the usually ineffective repressor to bind to the operator and stop RNA polymerase from going through with transcription. Together, the co-repressor and the repressor stop transcription and no more gene products are made. Thank you so much for watching this Kaplan MCAT science review about control of gene expression in prokaryotes. We hope it helps you with your studies.